it's it's scary, but it's also scientifically exciting to be looking at this particular virus.
All right, thanks.
Sarada, you want to ask our
you go ahead. It's an audience question, Lauren. Why don't you ask that?
Well, yeah. So Arad, can you unmute yourself and we will call on you? Okay.
If Arad can, I can state the question.
Here he comes. Here he comes.
Oh, good. Perfect.
Can you hear me?
Yes.
Yeah.
So I believe the first plot that Dr. Shokat showed with the MPA treatment, it looked like the protein levels of NSP14 also kind of went down. I'm curious if you think that maybe IMPDH2 might potentially be regulating the stability of NSP14 itself.
Yeah, great catch. That is what we think. I don't know if, anyways, yeah, it's pretty clear, like you saw from that slide 16, that when we add mycophenolic acid, the NSP14 drops. And you might be familiar, IMPDH has this very filamentous structure depending on the guanosine, the GTP levels in the cell. So that is one thing we're definitely thinking, that maybe NSP14 associates with IMPDH and is stabilized by that interaction. And that's how it, and then it also participates. And one neat thing is if IMPDH makes guanosine, then that's it getting methylated by the N7 side. So it's, that physical interaction is something more important than for stability too. Great catch.
Thank you.
That's awesome. Okay, audience, ask more questions. Until then, let's try something a little different. So let's try to brainstorm real fast and help you guys. So this absolutely mind-blowing result about that the effect of NSP14 on MHC expression levels is coming from the RNA methyltransferase activity. So do you guys have, I mean, you say you didn't know what was going on. Do you have an inkling of what's going on? Like, so I'll just toss out, I'll toss out a straw man. So do you think that it is, okay, first straw man. Do you think it is that precise enzymatic activity that's doing it, or do those enzymes have side reactions that might be doing the thing?
Yeah, like a neomorphic function that is somehow, one thing I don't think we showed is that connected to sort of Arad's point is that when we make the point mutation in the methyltransferase, it seems to lessen the interaction with NSP14. And so we think that also leads to destabilization. So it could be that sort of indirect thing again. And we could have been a little bit fooled, something about enzymatic activity and structure. Yeah, that's a wrinkle.
Oh, so how about maybe making some other mutations that aren't in the active site, but are in the protein-protein interaction?
Yeah, that's what Clem and the structural biology group is trying to solve, the IMPDH2 structure with NSP14. Actually, I was messaging him on Slack when Nevin was talking, asking where we were on that. And he said they've been making IMPDH mutants that have more stable structures to get the structure of the 14 so we could make those interaction mutants. Yeah. Nevin's group's got some really great sort of structure prediction ways to figure out what are the two surfaces that are interacting. It might be good to apply it there.
Yeah. Okay, but so for part two, I'll make the counterpoint. So I was looking, I just pulled up, some articles on N7 RNA methyltransferases. And it seems like one of their favorite targets are mRNA end caps. And so one could imagine that, some charge on steric alterations there would affect docking to the ribosome. You've shown that it's post-transcriptional, but it could be translational regulation. So,
wow.
How about maybe doing some in vitro translation assays?
Just spike in.
Mm-hmm.
Wow, that's a good suggestion. We have those assays going for a different project. So we could just put in NSP14 and the different mutants and see on a vanilla mRNA. I like that.
I guess also in the spirit of Lauren's brainstorming here, I'm just thinking about one of the 14 antibodies, Kayvon, is one of them is against NSP14, and we're working with a collaborator in San Antonio that can genetically manipulate the virus, and we're working with them to put in that mutation into the virus, Kayvon's mutant, and then what we could do is pull down in infected cells with the antibody, the wild type and the mutant, and just to see, just to get a little bit more insight into maybe what that mutant is doing. So we could talk to Lorena and we should set that up, Kayvon.
Yeah, I think we're trying to get that meeting set up right now.
So the obvious question would be, how do you think this is specific to MHC class 1? And if so, how could something like this plausibly be restricted to, that small a subset of genes?
Yeah, no, that's a good question. I mean, we checked other cell surface things, and they're not downregulated. So it's, to the extent that we checked a few things, it's not just globally sort of shaving off membrane protein integration. But that is, again, that's what the power of the virus is. So now the question is, we've got to design a better experiment.
Have you dug any deeper? Is it like, is it A, B, or C, or?
I forget. In fact, I think we checked, and I think it's all three, but HIV does something interesting, which is when it knocks the HLAs down, it knocks down HLA A and B, but leaves C up so that natural killer cells don't kill it. And C is the least highly expressed on the cell surface. So HIV selectively sort of left the one up to prevent natural killer cells, but downregulated the most of expression that would present viral. So I think, I actually can't remember if we did that experiment. It's another good one.
Yeah, and I think there are T cell lines and NK cell lines that express, preferentially express different subunit combinations. So.
Yeah, we've got some that are null and we can express whatever we want and things like that. T1 cells, T2, I forget.
This is just so mysterious.